Okay, welcome back to part 2 of this module. We're going to jump actually straight into a video just explaining what DNA and genes are. A little bit of this will be recapping what we did in the last video, but it's also then going to start talking about some stuff we're going to cover in this video. So get started with that now.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
Alright, so we're going to start talking a little bit less about actual structure, a little bit more about organization, and then we're going to get into actual expression of our genetic information. So there's a few terms we need to go over to help us understand how our genetic information is actually arranged and organized in our cells, in our bodies. So one of the first terms we need to look at is genes, and genes is probably a word you've heard before, and I don't just mean the type of pants, but genes are these sections of DNA. So you have your really long DNA molecule, and they can be broken up into smaller sections along there, and those smaller sections are genes. Now, what defines a gene? Well, a gene is going to basically be a section that has instructions on how to make a particular protein or carry out a specific function. So generally speaking, we would say each genetic instruction that codes for some sort of trait, be it eye color or something like that, that's a gene. So it's not just any old section of your DNA, it's actually a section that has a specific function. It does something in particular, it carries instructions for some specific sort of thing. Now, another word you might hear that sounds a little bit similar is genome, and the genome, the definition of that is the total set of all the genes within an organism. So we might talk about the human genome, or a rabbit's genome, or a strawberry's genome, and what it means is for that whatever organism you're talking about, what are all the genes that it has? And so what are all those specific sections of DNA that have certain functions that code for specific things that occur? That's what a gene is. One way you could think about it is your DNA molecule might be like a really big recipe book, and each recipe in a recipe book is a set of instructions on how to make a certain thing. And in DNA, if the DNA's molecule, the whole molecule's your recipe book, well, those genes are the individual recipes. They're specific instructions on how to do a certain thing. So that can be a really handy way of thinking about it. So your DNA molecule can have lots of genes along one molecule's length, just like a recipe book can have lots of recipes in it. Now, each DNA molecule forms a larger structure though. So one whole DNA molecule forms something called a chromosome. Alright, so one DNA molecule generally equals one chromosome. So if we go back to that recipe book analogy, your chromosome is the book. It's the recipe book itself. Alright, and then in there is all the different recipes, or in the chromosome are all the different genes. And we find the chromosomes in our nucleus of our cells. So you can think this way. Here's a person. If you zoom in and look at one cell, you can zoom in a little bit further, look at the nucleus, and in that nucleus you'll see chromosomes, these individual DNA molecules, and if you zoom into them really close all the way down, sort of as far as you can go, you'll actually see the DNA, that sort of twisty ladder that we were talking about in the other video. Alright, so that is how, generally speaking, our genes are organized into chromosomes. So that's one level of organization of the information in our cells. However, our chromosomes, those DNA molecules, are actually incredibly long. like really, really long. You're talking a couple of meters of genetic information in every single cell in your body. And you have that question, well, hang on, if it's that long, how do we fit it into something as small as the nucleus of a cell? And that's got to do with the fact that our chromosomes might be really long, but they're also really thin. They're like really, really thin, fine thread. And so what we do is, or what our cells do, is they actually coil them up, and they wrap them up and up and up into these tighter and tighter and smaller structures. And that's one way that they can help manage all that information in the nucleus. So each chromosome ends up being one very long DNA molecule that's wrapped around these specific proteins. And you can see here we've got some, they're called histones, and when you've got these little the DNA wrapped around histones, it forms something called a nucleosome, and then that coils up, and then that coils up into a supercoil. So it's like you coil something up into one sort of set of loops, and then you take that set of loops and you turn it into another set of loops, and then you take that and you turn it into another set of loops, and you just keep winding it up smaller and smaller and tighter and tighter. And that's how we actually manage to package these chromosomes. But not all the time. Sometimes they're unwound, so we can actually read the information in them. So as I mentioned, chromosomes are these really long coiled structures of DNA. They have lots of genes along them. And one thing to note is wherever those genes are in a chromosome, if you have another chromosome of the same type, so if you like chromosome number 1, if you have another chromosome number 1, it's going to have the same genes, and they'll actually be in the same position on that chromosome. In fact, that's called a locus, or loci, if you're talking about more than one gene. Think locus like location. So the physical position of a gene is its locus, which is just, it's like a fancier, shorter way of saying the gene's location on the chromosome. And so you can see in our image here, if we just were to draw our chromosome, it's this sort of like long stick sort of thing. These bands, these coloured bands, might represent our different genes, and there's lots of genes in different positions. If this was, say, chromosome 1 and I had another chromosome 1, it would have the same genes in the same positions. A little bit like if you buy, it's like having a recipe book that has, certain recipes in certain chapters, and you've got the same recipe book, you'd kind of expect to have those recipes in the same position throughout the book, that sort of thing. So you might be thinking, okay, that's great. I understand that DNA has these sort of small things called nucleotides that have these nitrogen bases in them, and we're interested in the order of those bases, and that's a gene. But how on earth does that actually, give me brown hair or blonde hair or blue eyes? How does that work? How do I get from these letters to that actual trait? And it comes down to proteins. You see, each gene codes for a specific protein, and it's those order of those bases, the A, the T, the C, and the G in the gene, that's going to tell the cell how they actually need to make that protein. And we'll get into exactly how that happens in a moment. But basically, your DNA, its whole job is to be instructions for making proteins. Now, those proteins, there's a whole range of different proteins. Protein isn't just like the same thing as meat. I know sometimes we talk about meat being a protein, but proteins is a very big category of molecules that has lots and lots of different things in it. So proteins are really essential for cells to survive, grow, and function. All right. And the way we get from our DNA to a protein is through two processes. The first one is called transcription, and the second one's called translation. Now, you may notice in the middle here, we have something called RNA, and RNA we'll get into a little bit later, but it's very similar to DNA, and it's kind of like copying paper or tracing paper, where you're able to make a copy of the DNA, so it doesn't have to leave the nucleus. But we'll jump on to that in a moment. First, let's pause and talk a little bit more about proteins, just to clarify what we mean when we say proteins. Like I said before, there's lots and lots of different proteins. In fact, you can see a link at the bottom here where you can go and learn about a whole different range of protein types. But three kind of common ones that we're going to talk about here are proteins that are structural. So that could be things like collagen, which you would find in tendons and ligaments. It could be keratin. Keratin makes up your hair, your nails, and in fact, in your skin as well. So those are like structural proteins. But then we also have enzymes. Enzymes is a type of protein that helps certain reactions occur faster. Amylase is a protein enzyme that helps you to digest and break down starch, and lactase is one that helps you with breaking down lactose. In fact, if you're lactose intolerant, generally what that means is that your body's not producing lactase anymore, and so you don't have that enzyme to break down the lactose, and that gives you an upset stomach. So those are two kinds, and then a third kind you might be talking about could be signaling proteins, and these are things also like hormones as well. So you've got something called epidermal growth factor, which if you cut yourself, that's a protein that needs to be activated to help tell your cells, your skin cells, that they need to start making more of themselves so they can heal the wound. Or insulin is a hormone, it's a protein as well, and it has to do with controlling your blood glucose levels. If you're diabetic and have type 1 diabetes, you may not actually produce insulin, and so you might have to artificially inject it into yourself. So these are just some types of proteins. Like I said, there's a link at the bottom of this slide that you can go to, and you can actually learn about a whole range of different protein types, because there's more than just these three. These are just some of the categories that are out there. So let's talk about how we actually get to making a protein. And now I mentioned before that there were two main stages to get from DNA to protein. And so those stages were called transcription and translation. Now there are certain enzymes that are actually involved here, and they're really important. They basically are things that will do the job of like reading the DNA and making the copy, and then they'll read the copy, and then they'll actually deliver the different components of the protein to be constructed. So there's some really important enzymes at play here, but generally this is the process we're talking about. We start with DNA, that gets transcribed and a copy is made, and the copy is in the form of something called RNA, specifically mRNA, and then from that copy is taken outside of the nucleus and it gets translated and converted into a protein. And that's sort of the general overview of how we make a protein from DNA. But we're going to dig into that in a little bit more detail. So let's start with transcription. Now, DNA is really, really important. If the DNA gets mucked up in any way, that's really bad for that cell. So DNA is too important to leave the nucleus. So we don't make proteins in the nucleus, we make them outside of it. Somehow we have to get the instructions from the DNA that's in the nucleus out of the nucleus to where the protein's actually going to get made. And that's where this first stage of transcription comes in. And if you actually think about the word transcript or transcribe, in fact, you'll get an idea of what's happening here. We're taking a copy, you've got one thing that's written out and you're transcribing that onto something else. So what we end up doing is we end up making a copy. Proteins made outside the nucleus. So instead we make a copy of DNA, and that whole process of making this copy is transcription. And you can see in the little video that's playing down the bottom, that's what's happening here. You've got this DNA going into this purple blob. That's a specific enzyme that's going to be reading it, and that moves along the DNA until it gets to what's called a terminator region, which is where it will terminate or end and finish reading. But what's happening is this enzyme, as it's moving along, it's reading that DNA and it's making a copy, this red copy of messenger RNA, and that's the stuff that can actually leave the nucleus. So you can see here it's opened up the DNA so it can see what there is. It's reading one side and it's making this new little sequence with a red backbone instead of a blue one, and that's your mRNA. So we can sort of simplify the process into these sort of stages. First of all, the DNA is unzipped because it's a little bit like a zipper. So it's opened up and it's only done at the gene that they want to copy. You don't need to do the whole thing, just the section you're interested in. And then it gets copied by making a molecule of messenger RNA. RNA is a molecule, like I mentioned earlier, that's really similar to DNA. And this RNA specifically is being used as a messenger. It's taking a message from inside the nucleus to outside the nucleus. So we call it mRNA. And we do that because RNA gets involved in this process in a few different stages, and there's a few different roles it can do. So we use the little letter at the front to tell us, oh, we're talking about messenger RNA. And it's going to make a complementary strand just to one side of the DNA. It doesn't need to do both, it just needs to get the instructions from one side. And so that mRNA molecule, that's just a copy. It's not that important. So it can leave the nucleus and it can go outside to where it needs to be to help make that protein. So this is sort of what we're talking about here. We've got our DNA at the bottom. And it goes A, C, C, A, T, C, A, G, T, C. And then the RNA is making its matching copy at the top. Now you might notice that there is a difference with the RNA, and that's because it's not exactly the same as DNA. In fact, on the next slide, we'll actually get into the two key differences that we have from RNA to DNA. So before we go any further, we want to make sure we understand how RNA is similar, but also how it's different to DNA. So there are two key differences. And the first is that the sugar is different. DNA uses the sugar deoxyribose, and RNA uses the sugar ribose. And the difference here is the sugar in DNA compared to the sugar in RNA is here, it's missing an oxygen. In fact, that's what the deoxy at the start means. It means without an oxygen. So it's a ribose sugar with one oxygen removed. That's what DNA has. RNA has that ribose sugar and it hasn't had anything taken away from it. So here you can see it's just an H, whereas here it's got an OH because it's still got the oxygen. So first things first, the sugar is different. The second thing that's different is the bases. And really it's only one base that's different. See, in DNA we have a C, a G, an A, and a T. But in RNA, we don't have that T. We don't have the thymine. Instead, we have uracil. And it basically, any place you would have had thymine in a matching DNA strand, you now have uracil. So you would write a U instead of a T. Now, that's really it for the differences. We'll go on the next slide. We'll actually have a bit of a practice at writing a complementary RNA strand. But some ways RNA gets used, like I mentioned earlier, it gets used as messenger RNA. Sometimes it gets used as transfer RNA or tRNA. And that basically, it's like a little carrier truck where it'll take an amino acid from one part of the cell and bring it to where it needs to be to help make a protein. So it's transferring that amino acid. So those are a couple of jobs that RNA plays. It does a lot more things, but those are just a couple that we're interested in at the moment. So let's take what we just learned on the slide before and let's put that into practice. Here I've got a strand of DNA and you can see what it reads there. What I want you to do in a moment is try and work out what its complementary RNA strand will be. Now, in terms of the letters, you won't notice any sugar difference, but you've got to remember that the bases will be different. So if you're writing out an RNA strand that matches this, you're going to have A, you're going to have G, you're going to have C, but you're not going to have T. So you can think of it this way. If you're making out a complementary DNA strand, then that first letter would be a T that matches with the A here. So it would normally be a T if it was DNA. But it's RNA, so it doesn't have a T, it has a U instead. So if you're writing a complementary RNA strand to this, it would actually begin with a U. Now, that's all the help I'm going to give you on this one at the moment. So what I want you to do is pause the video and see if you can work out the rest of the strand. And then when you're done, come back and I'll go through the answer with you. Okay, so let's see how you went and let's go through the answer now. So like I said, that first letter is going to be a U because instead of A matching with T, it's RNA, so it has to be A matches with U. So it's going to go U, A, U, G, C, A, U, C, G, C, U, A, U, U, C, C, U, G, G, A, U, C, U, U. So that's how it would work. Instead of basically, you could write this out like you were making it DNA and then just go through and replace all the T's with a U. Just remember as well, some people get a little bit confused here and they think that they have to match T's with U's. No, if you've got A here, then U will be on the other side. If you've got a T here, it's still an A here. Okay, that doesn't change. A doesn't get changed out. There's just no T's in your RNA strand. Okay, so now you've transcribed your DNA and you've made your mRNA copy. We need to take that mRNA copy and it's going to leave the nucleus and it's going to go to something called a ribosome. All right, ribosomes are really small structures that in a few different places in your cell, but basically your mRNA can go to a ribosome and the ribosome will read it and start making a protein from it. Now, we call this process translation because if you think the mRNA is kind of in one language, and that's the language of RNA, and we need to translate that into the language of a protein. So we're translating from sort of one language into another here. So the mRNA gets read, and it gets read in sequences of 3 nucleotide bases, and those are called codons. So you don't read it individual sort of letter by letter, you read them in sets of 3, like you're reading 3-letter words as you go along. And the specific term for that is codon. So you can sort of see that's what's happening over here in this image. So each codon, each 3 letters codes for 1 amino acid. Okay, now before I go much further, I do want to talk about how we actually make proteins, or what proteins are actually made up of, because you need to understand that to understand how we're going to get to there. So proteins are these big, long chains of amino acids that are all joined together. So we have these big long chains of amino acids joined together, and the way we get that chain of amino acids is by reading the mRNA. So, like I was saying, each codon codes for an amino acid. As your mRNA gets read more and more, you end up bringing in more and more amino acids, and they join together to form the protein. Now, the little animation here, which has stopped playing, was actually showing that happening. So I'll go back and get that to play again. So you can see that these are being brought in, and these little circles are our amino acids, and they get joined in, and they form this big long train that's moving off there, and that would become a protein. So we're actually going to practice making our own protein. So we're going to use the table that's on the screen here, and we have an mRNA strand up the top. So you can see it goes U, A, U, G, C, A, U, C, G, C, U, A, and onwards. All right. What you need to do is you need to break this into 3-letter segments, and that's your codons. So UAU would be codon number 1, GCA would be codon number 2, UCG would be codon number 3, and you can do the rest of it as you go. Once you've done that, read each codon. And so we'll start with UAU, and I'll help you out with this one, and then you can go away and do the rest. But UAU, we want to find that in the table. So if you actually look at this, on this side, you've got first letter, so the rows here are the first letter. Then you can find it by column, we'll give you the second letter. And then on the side, the third letter this is telling you individual rows in each box. So it's, the top one always ends with a U, the second one always ends with a C, the third one ends with an A, and the fourth one ends with a G. So we have UAU, so we're going to find first letter is U, so it's somewhere in this top row. Second letter is A, so that would be this column, so in this box here. All right, we've got UAU, and so we're going to, third letter would be the top one, UAU, there it is at the top, and both it and the one below it code for an amino acid called tyrosine. So tyrosine is going to be our first amino acid, and so I'd write that in, I write tyrosine, and then I'd go look at my second codon, which is GCA, and I'd find that and I'd write that in all the way through to the very end. All right, now some codons are stop codons, so they'd be at the end of a gene, and that tells the ribosome, hey, we're done, you can stop reading me now. And sometimes you'll also have a start codon or methionine, and so that's telling the ribosome, hey, this is starting point, this is where you need to begin. They may not actually occur in the sequence, you can just think of this as a sequence that's been plucked randomly from the middle of a gene somewhere. Alright, so pause the video and write out the rest of the amino acid sequence using that table. So go and do that now. Okay, let's go through the rest of those amino acids. So we already know that the first one is tyrosine. The second one, you should have gotten alanine, then serine, then leucine, then isoleucine, then proline, then glycine, then asparaginine, and then leucine again. So that is the sequence you should have gotten. If you didn't, maybe just go back and check that you got your codons spaced out correctly and that you looked them up correctly in the table. All right. So we're basically at the end here. We've got one more video to watch, just describing that whole process of going from DNA to proteins.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, now we're going to do a bit of a self-check quiz. You can see there are some questions up here. Now, depending on when you're doing this, you may also have a quiz where you can enter the answers into. So I'm going to read through the questions, but I'm going to make a separate video where I go through and explain the answers. So you'll need to write these down and then go find that video to check yourself. And remember, if there is a quiz that you need to enter them in, make sure you put your answers in there first. So 7 questions. Question 1, what do the letters DNA stand for? Question 2, DNA is shaped like a what? 3, the 4 bases, nitrogen bases are adenine, something, something, and something. 4, adenine always bonds with what? 5, cytosine always bonds with what? 6, DNA is important because it determines your physical what? And 7, nitrogen bases are paired together using hydrogen or covalent bonds. Alright, so go and answer that, and then come back and find the video with the answers and make sure you're all correct and you understand the answers as well.